Welcome to my video explaining step 2 of photosynthesis. So this step can be called the dark reactions, but make sure you understand that it's not because it happens in the dark, it's because we just don't need light for this to happen. So we went through the first step in the last video where we took in light, we took in water. This was called the light dependent, which means that it relies on light. It created a bunch of ATP and NADPH for us, and also oxygen that was a waste product. So now we're doing the dark reactions, but it also could be referred to as the light independent, which means it does not need light. Okay, we can also call this, rather than calling it the light independent or the dark reactions, we can also call it the Calvin cycle, because Calvin was the scientist that studied this and named it after himself, of course. Sometimes it's also referred to as the Calvin-Benson cycle because Benson was Calvin's sidekick. I normally just keep it sweet and short and call it just Calvin cycle if I'm talking about that. So in the light independent reactions, we're taking these two energy storage molecules, ATP and NADPH, to power this process. We're taking in carbon dioxide from the air, and in order to do this process, we have energy from these two molecules, and the whole point is to make a bunch of glucose or sugar. Okay, so this is the end goal of photosynthesis. The first step is just to make up energy, and then the second step is to actually make that energy. So we're going to go through the first step of the Calvin cycle, the dark reactions or the light independent. So it's called carbon fixation. So it's going to be taking carbon and we're going to be fixing it to another molecule that is called ribulose biphosphate. I like to call it RuBP for short. So I want you guys to understand that this happens in not the thylakoid membrane anymore, which was the first step of our photosynthesis. This happens now in the space of the chloroplast. It's filled with liquid. It's called the stroma. Okay, so in this process, we're taking a one carbon molecule, carbon dioxide, it's called CO2 because there's only one carbon. I'm going to use circles to represent carbons in this video. So I have one carbon, that's my carbon dioxide. Okay, all of the other elements, so the oxygen I'm going to ignore, the hydrogens I'm going to ignore, but they are going to be present in some of these molecules. But just to keep it easy, I'm just going to be referring to the carbons. So I have one carbon, and my RuBP has 5 carbons. So you can draw this picture at the bottom of page 7 and then fill in the steps as we go at the top, kind of halfway between the middle of page 7. So the space at the bottom of page 7 is for this picture. So carbon dioxide plus RuBP, 1 plus 5 is 6. So I have a 6 carbon compound now. But unfortunately, because I also have other things in this molecule, not just carbon, this 6 carbon compound is unstable. Okay, so its electrons are not properly made. It doesn't like to be together. It'd be like if I stuck a bunch of extra people in a van that only had 5 seats. Okay, it would be really, really squishy in there. So immediately, instead of staying as 6 people in one vehicle, they split 3 and 3 into 2 separate vehicles. So that is the very first step, carbon fixation. So it's going from these 1 plus 5 to 6, but it's unstable, so it goes 3 and 3 immediately. So that's the first step of our process. So I'm going to move into the second stage of our dark reactions, or our light independent reactions, or the Calvin cycle, which is called reduction. Okay, remember we talked about this word reduction. Reduction means that we are going to be adding things together. In the process, we are gaining electrons. So remember, G-E-R, gaining electrons is reduction. So we're adding things together. We're taking this 3 carbon compound and we're energizing it. So we're taking our ATP that we made in our first step of photosynthesis, and we're going to steal the energy from ATP to make ADP. So now we're taking off that phosphate. Okay, we're also going to take the NADPH and steal the energy from that as well to make NADP+. Okay, in the process, when we energize these things, we are now left with our 3 carbon compounds and a phosphate on the end. Conveniently, this phosphate came from breaking my ATP into ADP, and we now call it this really, really long drawn-out name. I don't even know how to say it, so let's just call it PGAL. Okay, PGAL is my name for the energized 3 carbon compound. They put that phosphate on it as a placeholder because, again, remember, we're trying to make stable molecules. We just added a bunch of energy to this molecule, and it's unstable when it's energized, so we put this phosphate on as a placeholder. Okay, in the meantime, we're going to have one of these 3 carbon compounds with the phosphate placeholder. It's going to be moving into a circle to replace my Ruby P, but that's in step 3. And the other one, my 3 carbon compound with the placeholder, is going to be used to make glucose. Now, if you remember, glucose has 6 carbons. It's C6H12O6. Remember, we're not worrying about the other stuff. So I'm actually going to take 2 of these down here. I don't need that placeholder anymore. Okay, the phosphate placeholder. I'm going to take 2 of these. I'm going to add them together. And I'm going to end up, because it's 3 plus 3 equals 6. Once I have this 6 carbon compound with the proper hydrogens and oxygens as well, now, conveniently, I have some glucose, which is literally the whole point of this process. Okay, whole point of photosynthesis is to make this molecule. Glucose is life. So that is reduction phase. Adding things together, these 2 things together to make glucose. I also am adding this with the phosphate, which would also count as reduction as well. Okay, and I'm using that energy that I made in the first step in order to do this whole process. Okay, the last step that I have is now making sure that I replace what I used up in order to make sure that next time I do this process, I have enough of the right molecules. So this is called replacing Ruby P because I'm literally just replacing it so that I have it for next time. It's like if I take out 5 eggs from my egg carton, and the next time I go, I need 5 eggs, but I don't have enough left. I have to go to the grocery store and replace my 5 eggs. Okay, so what I'm doing, remember I made glucose, I'm left with replacing Ruby P, so I'm taking that last PGAL that I haven't used yet. I'm going to take that, which again is 3 carbons. I would have to actually use 2 of these PGALs, okay, take off the phosphate, and because I only need 5 carbons, I would actually only need 5. So I have 2 of these because I need at least more than 3. And then I'm going to cross out this 1 because I don't actually need 6. I only need 5. In this process, I'm going to, again, break down some of my ATP in order to allow myself to have energy to break bonds and build bonds and that sort of thing. So this 6th carbon, okay, I'm not going to really do anything with it. It can be used again. If I do this process 6 times, I have now another 6th carbon, or 6 carbons that I can make some more glucose. So again, I'm going to just leave it there. It'd be like if I need a recipe and I have only 2 eggs in my recipe and I have 12 eggs in my carton. Do I have to use all 12 eggs? Well, no, I can save them for something else that I'm making. Maybe I'm going to make some scrambled eggs or some more cookies or whatever else that I use eggs for, and I can leave them in the fridge for later. So this 6th carbon here, I'm just going to save for later, and I can use it in another process. So this is just making sure that I'm ending up with the proper amount of carbons in the end so that this Ruby P molecule is replaced and I can go through this process again. Now, in class, we talked about how I don't actually balance things. In reality, this would be balanced. So I would actually start out with more than 1 carbon dioxide. I would start out with more than 1 Ruby P. I would be making more than 1 glucose. So I have a little diagram of this. And I would end up with sorry, it doesn't explain what I'm talking about, so I'm just going to go back. I would end up with more than one of these things happening at once, so that's why it doesn't really matter that I'm not using this 6 carbon, because it can be used in some other place, and it ends up working itself out. Okay, but because it's a complicated process, I just want us to know kind of with 1 carbon dioxide, what does it get me? And it gets me 1 glucose, but in reality, I would actually be using more than 1 carbon dioxide. But we don't need to worry about so much the numbers for this, because then it just brings in another thing that you have to remember. I'm trying to keep it nice and simple. So that is actually all for the dark reactions or the Calvin cycle. So you'll probably have to draw this out a couple of times. If you have any questions, please let me know. You're going to be drawing this in class tomorrow. So please make sure that you know it really, really, really well, so that when I put the 2 steps of photosynthesis together, you're not super confused tomorrow. If you have any questions, let me know. But if not, I will see you tomorrow, and have a wonderful evening. Good night.